Good morning, good afternoon, everyone. Today is an important event for the OpenScope program as a whole. Um, every year we have we release a data set that we collected in previous years, and this event today is meant to introduce the data we will release later this year. But before I start, I'll give a little bit of an introductions about the OpenScope program as a whole, as well as provide some context for the presentations today. So first, I want you to keep in mind that the OpenScope program has a large team behind it, and so we are all incredibly thankful for the work that they do. First, a large number of scientists, research associate, managers, engineers, and animal care teams have dedicated a lot of effort and time to collect the data set that we present today. The project that we will present were selected by reviewers, anonymous reviewers, as well as the OpenScope Steering Committee, with the people that are listed on these slides, and I'm also incredibly thankful for their work. They are the reason for the success of those projects, and so it's important to highlight them. The project is funded by the Brain Initiative, by a Brain Initiative grant that is listed here. They took the bold initiative to sort of select this exploration into social engineering of science, and so I'm incredibly thankful for their support. At the bottom of the slide, you see a number of leaders that influenced the programs over the years, and it's thanks to their work that we are here today. So, what is OpenScope? We are an observatory of the mind. This is a picture that was taken from a paper put together by Christoph Koch and Claire Reed more than a decade ago. Their vision was that neuroscience will largely benefit from collecting data set that impact the field as a whole, and eventually provide those tools as how they are used in other fields such as astronomy or particle physics. What you will see today is that we essentially realize that vision, and moving forward, we hope for this model to grow. In the last 10 years, the Allen Institute has collected really influential data set using neuroscience pipelines with awake animals, in this case, mice. What I'm highlighting here is two important studies on the left using 2-photon calcium imaging in mice, and on the right using neuropixel electrophysiological recording in the same animals. We recorded similar visual stimuli across both species, both experiments, and provided this data set to the community as a whole. Hundreds of animals were used, and the data set were reused by the community. So first we had our own scientific questions, and so those studies highlight the answer that we found in those data set, and later on, those experiment data set were reused by the community. You can see on this slides on the left, this is an analysis that was done in astronomy for data sets that were generated by the Keck Observatory more than 2 or 3 decades ago. The time it takes for those studies is in the order of 5 to 10 years, but the number of publications that come out of this platform rise over the years. And so you see that this has a potential to transform how the community study its object of interest. On the right, this is an equivalent plot that Saskia de Vries, Josh Siegel, and Christoph Koch put together on our own dataset, the one that I mentioned earlier. And you see that from the data release, approximately 5 to 10 years later, the number of publication rises. And this is a testament to the value of sharing and providing data reuse for the community. This is core to a principle for the OpenScope program, and this is why we have this event today, is because we see that in the future, those data set will become anchoring point for the community. They have a value of their own beyond the initial design that the team will introduce today. So OpenScope is a community observatory in neuroscience that try to implement those principles. So first, worldwide scientists, and today you'll see that we truly represent the world. We have teams across the world, from Israel to Canada, and some in Germany. So we're sort of really receiving projects from all over the planet. They propose projects uh, to a platform, and we run them with our staff. OpenScope operated with either 2-photon or NeoPixel recording technology. And our goal is first to maximize our investment into the platform by making it available for new experiments. And then we want to sort of, we aim to help build and support a community around it. And you'll see today that, I hope you'll see today that we achieve that. And to eventually help the system neuroscience ecosystem to develop and help projects sort of integrate better with one another. So that's our goals. To achieve those goals, we have a number of components in the OpenScope program. First, this is the key aspects, so access to our end-to-end data collection pipeline. So teams propose projects that are run on a system where we have surgical procedure that are standardized, our neuronal recordings that are standardized, as well as the data packaging that is part of it. Every year in the last 5 years, we run a proposal cycle with double-blinded review. So this is to remove any potential bias in the selection process, and it's open to the entire world community. Then, once the data is acquired, we'll leverage a cloud data deployment and analysis ecosystem. So on the bottom right, you see what we see as collaborative project, a sister project to ours, the NWB, the Neurodata Without Border, which is a standardized file format that we leverage, and the Dandy archive, which allow us to share those data set with everyone quickly. The last component of the OpenScope program is the technical hub. So every year in the last few years, in June, we run a technical workshop, which you'll see examples in the later slides. This year, because the program is at the last year of its current funding cycle, we'll not have this event, but I'm hoping that in the future we can reinstantiate it. And the last component is this event today, which is the scientific a scientific event where we disseminate the data set online. So what have we done? So in 2023, we released 3 data set. One from what we call the dendritic coupling project, the second one from the global local oddball project, and the third one we call an illusion project. This was from team distributed in Canada, one in Berkeley, Yi Young-shin, which moved to Korea, and then a team at Vanderbilt. 2 of these studies have now been either on archive or published. One of these in Nature Neuroscience last year. So we are making progress on that front. Last year, we released 3 data set, one from the temporal barcode project with Pamela Renagle, another with Michael Berry on temporal sequence learning, and a third one with Chinmay Puradare and Mayank Mehta at UCLA, and on the we call the Vision to Hippocampus project. All of those data assets are public, and you can access them on Dandy. The reason I'm showing those screenshots on YouTube is because the event we hosted in the last few years is also visible for you to see the overall context of the studies. And if you want to access the data, there is extensive documentation on how to do so. In addition, we also hosted the technical workshop that I mentioned earlier. And so you can see on our YouTube channel all of those recorded videos. So those provide important context to understand how the experiment is acquired in our platform. I want to highlight that this year we're doing a community project. This is ongoing, and so this is an opportunity for you to participate in the OpenScope program. So in a year ago, we organized a review of predictive processing with more than 60 co-authors. This study is on archive. I think it goes beyond the scope of this meeting to go in depth there, but we have a 100-page long review that highlight the state of the field in predictive processing that was written collaboratively. And in this paper, we actually describe an experiment that is ongoing now. And the data for this study is currently being released regularly on Dandy. This is the experimental design. Again, I invite you to go to the archive piece, and you can join us every week on Tuesday morning at 9 a.m. We have our team meeting, and all of those meetings are completely public. The entire process is free for everyone to participate. And so we continue to work on this OpenScope community project. So today we are going to work towards the release of all 4 projects. I'll mention the date in the next slides. I thought it was important to highlight that what we're doing is really important because we want to facilitate data reuse. It's really important for helping the community grow. It's really important to sort of limit the number of mice that are used across the field, but it also is an opportunity for collaborations across the field. But I think it's fair to say that people are anxious with sharing data in the community, and that's because the field is very competitive. Just want to highlight it. And as a unique platform, but try to establish the first observatory in the community, we want to promote the collaboration values of those data reuse assets. And so what we suggest is that if you're interested in any of these projects, please reach out and access the data set on the Dandy assets that when we share it, and then talk to the teams that put together this project and try to help them out on their project. We want to facilitate this collaboration, so it doesn't have to be done in a way that it feels competitive. It can be done in a very open and collaborative way, in a way that everyone benefit strongly from this work. The field has limited resources, and so we just need to synergize them. So here I providing a QR code with a Slack channel that you can join. So feel free to do that, and I'm happy to route people to sort of facilitate connections. In addition, you'll see the presentation today. Feel free to reach out to any of those people if you're interested in helping them out. Okay. So this is a program today. As you can see, we have 4 projects. It's a really busy schedule. In 2024-25, we selected 4 projects. That was our goal. The first is going to be presented by Julia Veit. This is a cool project with Henning Spreckler in Germany. The data release we call it the Vismo project. They will tell you a lot more about what's the intent of that study. The data release for that project will be in August 29, 2026. And this is because all of our projects have a 1-year embargo period. And 1 year after the team has access to the entirety of the data set with sufficient metadata is our policy on when we release it. It allows the team to sort of have a head start on their analysis because they put so much effort into it. And that's what we decided to do. The second project is will be presented by Roberto De Filippo and Torben Ott, again from Germany. We call it the Psychode project, and the data release for this project will occur on June 11 this year. The third project will be presented by Noah Barnea and Danielle Deitch from the Weizmann Institute in Israel. We call it the Loop project. It speaks about memory. The full data release will be on May 6th this year. It's from Yaniv Ziv Lab in Weizmann. The last project will be presented by Federico Bolanos and James Gardner with Javier Rolandi, which I think is here today too, from Canada. And it will be about texture discrimination. The data release for this project will be on June 17. And then we'll finish today with a few introductory code notebook that Carter Peen and the team put together, but in the intent of helping everyone get started with the analysis of this data. So with that being said, I'm going to hand it off to Julia Veit to start the first presentation on the science. Unless there are any questions in the audience. All right.
Okay.
Julia?
Yep. And I will share. Can everyone see this?
I cannot. Maybe I'm on the wrong screen.
Maybe the wrong screen.
No,
sorry.
I can't see it.
Let me try again. Is it working now?
Yeah,
I see a screen.
Okay, perfect.
Perfect.
Then, yeah, I also first want to give a huge shout out here before presenting our project Vismo. First of all, obviously to the Allen Institute and the amazing help they provided along the way in really guiding us all the way from stimulus development until now for the data handoff. And yeah, I mentioned a couple of people here. Oops, we interacted with more. Of course, Jerome and Carter we've been in contact with a lot, but there's many, many others, probably too many to name. And I want to say that this project is a team project between the labs of Henning Sprekeler at TU Berlin, where especially two grad students, Sophia and Saif, have been involved a lot in the analysis of the preliminary data. And Lorraine Herteg is on board now as a senior postdoc. And then my lab at the moment is kind of in the middle between the University of Freiburg and Bremen, and my postdoc Yael Oran is working. She was the person developing the stimulus and is now also very much driving the analysis. Okay, so this project, we are interested in basically visual motion, right? And we know motion is ubiquitous. As soon as we start moving our eyes or moving ourselves, the picture on our retina will be in motion, right? And this is sometimes very predictable if we're actively moving through our environment, and sometimes less so if we're maybe sitting in a train, but we'll have like a wide field of coherent motion passing on our retina. However, there can also be local motion that could be a sign of interesting things happening, right? There could be predators, there could be prey. They could alert us to interesting things happening. And sometimes we have kind of both types of motion on top of each other. So we have some global motion, and then a different motion happens on top of that, which would equally be a sign for something interesting happening in the environment. We know a lot about the representation of motion, especially from studies in primates, human and non-human primates, where we obviously have this very beautiful hierarchical visual system with these two these two very famous streams, the ventral stream leading into the temporal cortex, giving us the details of objects in the visual scene. And then we have the dorsal stream leading to these areas MT and MST, where neurons respond very selectively to visual motion. And neurons here are tuned to both the direction of motion, here with moving bars. You can see they're not tuned for the orientation of these, like the neurons in V1 are, but actually to the direction of motion. They're also tuned for the speed of motion. And over the years, they've been characterized a lot with these so-called random dot kinematogram stimuli, which consists not of bars or gratings, which have been used extensively for the early visual system, but of like these displays of dots that can move all in the same direction, which would be 100% coherent or correlated stimulus, or could vary where a certain percentage of these dots move in the same direction, whereas everybody else is moving in random directions. And it can be obviously zero correlation or zero coherent stimuli, where there is motion in the scene, but it's just not moving in a consistent direction. And here too, neurons in a primate MT are tuned to the coherence of motion. So here the open bars denote the preferred direction of the stimulus, and you can see how they really increase their firing here as the coherence of the stimulus is increased. If we're not only interested in these things, but also this local global divergence, we can see that there's actually two types of cells in primate MT, right? There's some that respond more to, so this is the stimulus where both the direction is always the preferred direction of the stimulus and the coherence is always full coherence. But as we increase the size of this display of random dots, basically, you can distinguish two types of cells, one that basically responds more to these larger wide field motion stimuli, whereas others are surround suppressed. So they respond more strongly to these small local patches of motion in the visual scene. And interestingly, at least in the primates, these seem to be organized into columns anatomically, right? Where you have specific bands in monkey MT where neurons live that have this more integrative, facilitatory surround, and then other interbands where you would have more of these neurons that are suppressed by these large wide field motions that respond more to local motion. So this basically gives a model of primate MT where you basically have columns with preferred direction of motion orthogonally organized with these bands and interbands of integrating and basically local motion sensitive wide field and local motion sensitive neurons. If you have these neurons that do respond more to these smaller patches, there's also an interesting finding that if we map the preferred direction of these, for example, this is one cell that prefers motion close to 0 degrees, which would be 0 here in this right plot, where then we put the motions in this round in motion as well. And if that motion direction is the same, these neurons are strongly suppressed. However, if the motion is very different in the surround, we actually get a facilitation even on top of the preferred direction. And this is another cell that shows a very similar tuning, this one preferring 210 degrees. And you can see that it's strongly facilitated, almost doubles its firing rate when there's offset motion in the surround. And this reminded us a lot of an effect that actually has been studied a lot in mice recently, but more in the primary visual cortex. And you can see this here in a static display of gratings, where you have two identical gratings here that you might perceive as not really being identical. In fact, the left one could look maybe a little fainter or a little less contrast rich. And this is probably due to the fact that we basically get very strong responses if a neuron's, if a stimulus, in this case, this grating, is about the size of a receptive field. But then again, if we put on a surrounding stimulus that has the same orientation in this case, so we're not in motion direction, but in orientation space, we get this very strong surround suppression, which could be the reason why this doesn't look as contrast rich. But if we offset here again, just in the orientation space, the surround from it, the response of this neuron basically bounces back to nearly values as for the center-only stimulus. And as I said, this has been studied a lot recently in mice. This effect also exists in primates and cats and all these model systems studied. But particularly in mice, because here we have the chance to look at inhibition. And we now think we know inhibition really seems to play a strong role, especially for this surround suppression. We know inhibition in the cortex is really a zoo of different cell types. And the Allen Institute has actually played a huge role in kind of clearing up that for us and really characterizing this variety of cortical inhibition, really. Here in this project and also in the previous work, we mainly focused on these kind of canonical interneuron subtypes, which are the parvalbumin cells, which are mainly your basket and chandelier cells that have this strong inhibition of the soma of pyramidal cells. You have your somatostatin cells, which have a large group of Martinotti type cells, which targets the dendrites, but also some non-Martinotti types. And then today, I'm only going to talk about the VIP part of the 5-HT receptor inhibition cells, which have been shown to selectively target other interneurons, especially somatostatin cells. And that means they basically form this disinhibitory motif, right? If VIP cells become active, they release pyramidal cells from inhibition by somatostatin cells. And here, what we know is that while pyramidal cells and also these parvalbumin interneurons become surround suppressed if we increase the size of a grading into the surround, somatostatin cells tend to not be so much surround suppressed, but show this more integrating property, where you would see them actually most active in cases of a large stimulus. And the hypothesis is obviously easily made that this strong inhibition by somatostatin cells could be important for the surround suppression of the other neurons. And indeed, if one optogenetically silences somatostatin cells, pyramidal cells become released from at least a part of their surround suppression, showing that SOM cells actually do play a role here. In the same study, it's been also shown that these SOM cells integrate from wider areas of cortical space, which kind of makes sense for a neuron that has these properties. In the case of the offset surround, really almost all pyramidal cells fire a lot more in this condition that I called the cross, right, where the orientation in the surround is offset. And here, if we look at somatostatin cells, you can see that they really have a distinctly different tuning, where again, they like the homogeneous or the same orientation in the surround a lot more than this cross surround, which again could be the reason why the principal cells are tuned this way. This effect, however, couldn't really be explained by their further integration, right? These stimuli have the same size. So the idea of actually a couple of labs over the last years have been that potentially VIP cells could play a role in this because they are also similarly tuned, like they respond a lot more to the cross surround. Meaning if they are very active here, they could be the reason why somatostatin cells are less active here, and then thus potentially lead to the tuning of the pyramidal cells. And this has also been tested using optogenetics. The Scanziani lab were the first to publish this. And there is a small but significant effect of reducing the surround modulation when suppressing VIP neurons. We've recently found that actually excitatory circuits also seem to play a role. So somatostatin cells seem to also receive more excitatory inputs in the case of the isosurround compared to the cross, which we verified with 2-photon holography, where we could show that basically somatostatin cells get more excitatory inputs from pyramidal cells that have a very similar tuning curve, so that prefer similar orientations, whereas if they prefer different ones, we actually don't see much excitatory inputs from these. Meaning interneuron circuits probably play a role, but also orientation-specific connectivity could play a role for mediating this. But we also still think this is still not the entire story, probably, because even phase offset surround. So here we do have the same orientation in the center and surround. And here the study from Wolfzimmer lab shows that there is a very similar contextual modulation also for this. And by some own preliminary unpublished data, we can see that here the effect is actually very correlated to the case of the offsets around, meaning potentially there's a similar mechanism that kind of generally detects these non-homogeneities in stimuli. And so this is where we thought, back to these monkey studies, whether motion stimuli, so where we basically take all orientation tuning out of the picture, could also lead to these contextual effects. But here we basically noticed that there really isn't that much data there, right? We don't know that much about motion processing in the mice. We know a little bit. There's a study from Petrianu lab showing that mice can detect these random dot kinematogram stimuli, that they have beautiful tuning curves if you vary psychometric curves, if you vary the coherence of these random dot kinematograms. And that also they showed by applying mucimol to primary visual cortex, that primary visual cortex seems to be necessary for this. We know that there's some higher visual areas in the mouse that seem to care more about motion than, for example, textures. We know the higher visual areas kind of represent not the entire visual field, and that these anterior higher visual areas in the mouse visual system that correspond to the lower visual field, where the mouse would see a lot of coherent, basically full screen, full field optic flow, could be more important. And indeed, then this story by Sid and Gord showed that these anterior regions correlate more to motion processing and are also more tuned to coherent motion. But in general, these studies are still kind of using different techniques, different stimuli, and it's kind of hard. And there's no study that really looked into the center-surround interactions particularly. And this is basically where we came up with this OpenScope project, where we really wanted to do this large-scale characterization of the representation of motion, especially looking at the direction selectivity, but also the coherence tuning, and especially these center-surround interactions for motion stimuli, right? Whether we see areas or cell types that have particularly reinforcing surrounds or these suppressive surrounds, and how these canonical circuits, right, these SOM, VIP, disinhibitory circuits, react in these cases, and how they could be differently tuned in different areas that might have more integrating surrounds than suppressive surrounds. So that's basically what we wanted to know about when we came up with this this project. Another thing we were really interested in is how stable this representation of motion is over days and weeks, right? In other words, is there representational drift? Does the preference or the suppressive or reinforcing surround change over time somehow? So the project we came up with is basically using the multiplane two-photon mesoscope of the Allen Institute, where we can nearly simultaneously image 8 planes across the visual cortex. We basically have this passive viewing pipeline of the Allen Institute where they implant the mice with windows and then do intrinsic signal optical imaging to identify the higher visual areas. Then the mice are habituated to the rig, and then they do their OFIS pipeline where they basically show these stimuli I'm going to show you in a little bit. And we did this in 4 different mouse lines because we were interested in all these different cell types. So we had 4 SLC mice that label pyramidal cells, and then 3 VIP mice, 2 parvalbumin mice, and 2 SST mice that were imaged in this way. And we had 2 sessions where we imaged 4 different areas per session in 2 planes, 175 microns and 275 microns, more or less. So basically 8 planes per session. In the first session, we kind of focused on these anterior visual areas as well as V1. And then we had a second session where we repeated RL and the primary visual cortex, but then instead of AM and AL had PM and LM imaged as well. The visual stimulus basically contained these random dot kinematograms, and you can see here that we had these global and these local motion stimuli. Local motion, I'm going to repeat it, being both this relatively offset motion where the dots in the surround move in a different direction, or only the local motion patch, and then compare this to global motion in all of these areas and all of these cell types. The other stimulus we had, or that's actually interspersed with this, is we do this at different coherence levels. So here again, we have basically a full coherent condition, which is what you saw in the movie up here. But we also have a condition where these dots are only moving at 60% coherence, and we have a fully 0% incoherent condition. In total, this gives a lot of different stimuli conditions. So we basically have 8 different movement directions in the center. All of these have a condition where the surround direction is basically matched, which would correspond to the global motion. All of these have a condition where we only show the center, which is the center only condition. And then basically for 4 different surround directions, we also have these varying levels of relative offset between the center and surround, which I tried to identify here by colors. So 180 would be the exact opposite motion. And then, yeah, there's basically the smooth variation away from the main motion direction. And then as we close the circle, we get closer again to the original center direction. And we do all of these conditions once at 100% coherence, once at 60% coherence, and then we also have both the full screen and the center only condition for the fully incoherent condition. So overall, this gives 92 different conditions that are shown for 1 second each, with half a second interstimulus interval. So these are the main meat of the project, let's say, but we also have a couple of other stimuli added in. Especially important, we have a couple of receptive field mapping Gabor patches. So these are the same that were used for the Allen visual coding pipeline. This is very important to see which neurons basically correspond, have receptive fields that correspond to the central region here of our moving dot stimuli. We also have a short session of drifting gratings, just so we can compare the strength of the tuning to both of these visual stimuli across the condition, across the areas and cell types. And we also have a homogeneous gray screen. I don't know what is grading here. This is no grading there. This is literally just a gray screen for baselining. Exactly. But then the largest part of the session is obviously the different conditions of the random dot kinematograms. And then to look at drift, we kind of asked them to do this, and I think this was also quite an ask on our side, to basically record the one session on a one day, and then another day record the second areas, and then basically wait a week and then repeat both of these sessions again, and then wait another week to repeat both of these sessions again. So this basically gives us then the chance to track the same cells. So the exact same fields of views were reused in all of these sessions. So we can basically track over time the different ROIs and see how the representation of these stimuli changes over time. And yeah, with this, we basically got a total of thousands of principal cells and VIP neurons and hundreds of SSTs and PV cells delivered. I think these are not really the numbers because this doesn't yet account for the repetitions of the same fields of use over weeks. So it will be a little bit less. I cannot, unfortunately, really tell you that much yet. We're still in the very early stages of actually analyzing this, but we are very, very excited to dig in, and we definitely have great responses. And yeah, if you're interested in this, definitely drop me or Henning a line, and we can prep you for this. And I want to just thank again the Allen Institute and definitely the group of Henning and Yael for being incredibly helpful for pushing this project forward. Thanks.
Thank you, Julia. It's a
Is there any questions?
wonderful presentation. I have a small question, and I will see if the audience I have some for you. The comparison with the primate is particularly interesting to me because we know we actually have lots of folks interested in this here. How did you consider the receptor field size difference between primates and mice in your design?
Yeah, this is obviously very hard, right? We know in the visual system of primates, receptor field size gets larger a lot faster than it does in the mouse. In the mouse, it increases maybe 10% or something with each step in the hierarchy, and there's maybe not even that many steps in the hierarchy. But yes, obviously, primate receptor fields are much smaller and much more local in the primate. However, these the orientation to surrounds that exists pretty much throughout all studied animal models, right? This has been found in cats. This has been found in tree shrews, in monkeys, different kinds of monkeys, and it's also there in the mouse. So even though, of course, the acuity of the mouse is far less than that of primates, we do think that this could be probably conserved circuits potentially.
Yes. Other questions in the audience for Julia? All right.
Okay,
If
I'll
not,
stop sharing.
thank you so much. It was wonderful. So next, Roberto De Filippo will introduce his own project. So please take it away.
Okay. Do you see the presentation?
Yes.
Oh, great. Okay. Okay, so I'm very happy to be here today and present our project about psychedelics that was selected last year. I want to first of all thank all the OpenScope team for working on this project and collecting the data, especially because it required some additional effort to implement some things that were not done previously, like IP injections. The project is called PsyCode, sensory coding under the influence of psychedelics. And specifically, we're talking about psilocybin. So why to study psychedelics? One reason would be that they are currently being investigated as novel therapeutics for depression. And this clinical interest has generated significant public and institutional attention. So this slide illustrates the current landscape, with major publications like the New York Times reporting on late-stage clinical trials of different substances, including psilocybin, and framing them as potential breakthrough solutions in psychiatry. This coverage reflects a convergence of academic research, investment interest, and shifting regulatory attitudes. And given how close we are to seemingly have widespread legally available psychedelics, you might think that we know a lot about what they actually do in the brain, but that's, however, really far from being correct. Okay, so now to be clear, our interest with this project is not the clinical application of for depression, because what we're actually interested in is understanding a more fundamental feature of these compounds, that is the mechanism behind the classical sensory distortions that define the psychedelic experience, visible here in this realistic animation. Yet the neural circuit underlying these distortions remains surprisingly poorly understood. So one thing that is certain is that 5-HT2A receptor activation is necessary to induce psychedelic hallucinations in humans. But exactly how the activation of this single molecular target produces such specific and peculiar changes in visual processing is still an open question. And that is the gap that we are trying to fill with this project. And this is also where the etymology of the word psychedelic becomes instructive. So we have a psyche, that means mind, plus the lane, that means reveal. So psychedelics as mind revealing. The term itself suggests that these compounds don't just alter experience arbitrarily, but they might reveal something about how the mind is organized. So the leading computational framework for understanding how the brain actually constructs perception is predictive coding. That is a specific implementation of Bayesian models of perception. So the core idea is that our experience of the world is not a direct readout of sensory input. Instead, the brain combines bottom-up sensory signals with top-down expectations about what the world contains. So for example, if you're trekking through the desert, you're much more likely to encounter a camel and not a polar bear. And your sensory system uses this knowledge to bias perception towards the most probable interpretation. So we see psychedelics as compounds that act on predictive coding circuitry to give rise to sensory distortions. Exactly how they do that, whether by modulating priors or prediction errors or both, is unknown. So here we are using psychedelics as probes to dissect the circuitry of perception. And by understanding how they distort perception, we hope to better understand how normal sensory processing is constructed. This figure here comes from a really recent review on psychosis, but it captures a central question in the psychedelic field perfectly. Because with psychedelics, just as with psychosis, sensory distortions can be explained within a Bayesian framework in a variety of ways. And the literature offers two main competing accounts. So let's start with the bottom-up or weak prior account. This theory suggests that in psychosis, the brain's internal predictions, its priors, are not precise enough. Because these priors are weak, they fail to filter out or suppress incoming sensory noise and internal signals. These failures make sensory data overwhelming and abnormally salient, which is thought to be the starting point for many symptoms. On the other side is the top-down or strong priors account. This proposes the opposite problem. So the brain priors are too strong and too precise, and these overly rigid expectations end up overriding and distorting the actually sensory information coming in, forcing us to perceive what we expect rather than what is really there. And this explains how false belief can persist and even shape what we see and hear. Now, here's why this matters to us. Psychedelic distortions could in principle arise through either mechanism as well, or some combination of both. Here, indeed, we can see 3 papers, each offering a different account of what psychedelics are doing to predictive coding circuitry. So on the top, we have the Rebus model. So this is the weak priors account. So the proposal is that psychedelics relax the precision of high-level priors, allowing bottom-up sensory information to exert greater influence. The brain therefore becomes less constrained by its expectations. In the middle, we have the opposite. So again, hallucinations and strong priors. On this view, psychedelics might actually strengthen priors or make them overly precise, causing perception to be hijacked by internal expectations rather than sensory data. And on the bottom, there is a third possibility. Here it is proposed that psychedelics don't primarily act on priors at all. Instead, they enhance prediction errors directly. So the brain is flooded with a fake surprise signal, and it's this aberrant salience that drives the perceptual distortions. Okay, but here is the problem, because how predictive coding might actually be implemented in the brain is still largely unknown. So these are computational theories, but not real circuit mechanisms. So they tell us what the brain might be computing, but not which cells or circuits are doing the computing. So the Rebus model, so in this slide, we can see that it does take a step in this direction. So it's a circuit implementation. It's based on a canonical circuit motif where pyramidal cells of layer 5 encode the priors, and superficial cells of layer 2-3 are encoding prediction errors. So in this model, deep layer pyramidal cells, which happen to express high level of 5HT2A receptors, are directly activated by psychedelics. This activation makes them more susceptible to ascending prediction error signals, and it's this process that's proposed to reduce the precision of high-level priors. Notably, in these models, there is no explicit role for interneurons, so the entire account is framed around pyramidal cells. But recent experimental results suggest that this might be an incomplete picture. Because a growing body of work points to a critical role for interneurons, and specifically somatostatin-positive cells, in predictive processing computations. Moreover, we know that pyramidal cells are not the only one that express 5HT2A receptors. So using this a recent whole brain MERFISH dataset from the Allen Brain Cell Atlas, so a spatially resolved single-cell transcriptomics method that allows us to map gene expression across cells while preserving the spatial location. We can ask exactly which cell types are positioned to respond directly to psychedelics. So the data here shows the cortical subclasses with the highest 5HT2A RNA prevalence. So we see high expression in deep layer pyramidal cells, as expected, of this graph. But there are also some interneuron subclasses that we can see here in blue. So we have both somatostatin neurons in light blue and parvalbumin neurons in brown that show clear 5HT2A RNA expression. So we can say that the receptor is not exclusive to excitatory cells, but interneurons are also in a position to respond directly to psychedelics. Okay, next I will show some experimental data pinpointing a possible specific role of somatostatin cells in predictive coding. So I will go through these papers very quickly because they're very complex, so there is no time to explain. So in this first paper, predictive processing was investigated with a virtual reality environment where visual flow was controlled by the animal locomotion, as seen in panel B. So here, visual flow was also controlled by the experimenter, so giving rise to either negative or positive prediction errors. So a negative prediction error happens when the visual flow is blocked, but the animal is moving. And the positive prediction error at the opposite happens when the visual flow is positive, but there is no movement happening. Okay, here the authors were able to identify somatostatin neurons as responsible for inhibition to neurons that detected negative prediction errors. So these neurons are here represented by the red triangles. And so these neurons are excited by locomotion, but inhibited by visual flow. And the inhibition by the visual flow is provided by this intermediate neuron that is a somatostatin neuron. Okay, in this second study, the authors also use a virtual navigation task, but here the animals run down a track and turn either left or right to receive a reward. So on some trials, the trajectory towards the reward is smooth, so the head direction doesn't doesn't need any correction because they just arrive at the end and they turn right. In this case, so here on the left, we also see, so on the y-axis, the progress of time, and we see the activity of a subset of somatostatin cells, somatostatin 44 positive. Then in red, we have somatostatin 44 negative, and then the other neurons. So in this first case, where there is a smooth trajectory towards the reward, we don't have any special activation of somatostatin 44 cells. But if we go in panel C, in this case, you see that the head direction is actually, there was a mistake at this point where they turned too much. And here you can also see that the somatostatin 44 cells are activated specifically at this time point. So the burst occurred when the mouse had turned away from its typical trajectory, resulting in a heading deviation, consistent with a corrective action to move back towards the typical trajectory. So this reveals a population of SST neurons that specifically encode navigation errors. In this third paper, the authors describe a mechanism in V1 that boosts sensory responses when an animal expectations about visual stimuli at specific locations in a virtual environment are violated. These prediction errors selectively amplify the representation of unexpected visual input through synergistic interaction between high-order thalamic input and local VIP to somatostatin disinhibitory circuits. So again, in summary, we can say that various recent experimental work showed that somatostatin neurons play a specific role in computed prediction error across different tasks. And we also know that somatostatin neurons can even potentially be directly activated by psychedelics via 5-HT2A. So our main question that we had with this project is to understand whether somatostatin neurons contribute to the effect of psychedelics on perception. Okay, so here is the paradigm for our project. So we decided to use to have active behavior. So the classic visual change detection task done already at the Allen before, plus passive stimulation that consisted of receptive field mapping and surround suppression stimuli. Here we decided for surround suppression because we know that somatostatin neurons are also involved in this circuitry, as shown in the previous presentation. Okay, here in the image, you see the structure of the session. We have a passive stimulation block, a change detection block. Then we have a break so we inject the animal with either psilocybin or saline, and we wait. So we wait because in our experience doing experiments in rodents with psilocybin, we noticed that after the injection, the animal tends to be not very active in a behavioral paradigm. So for this reason, also in this case, we are waiting. And after this break, so we start again with the change detection task, and then we have a new block of passive visual stimulation, and at the end we have optotagging. So with this structure allows us for a within subject comparison of neural activity and task performance. So we can compare the post-injection to the pre-injection epochs. So for recording neural activity, we went for a neuropixels probe. So we have 6 neuropixels per session, targeting visual and frontal areas. And for the optotagging, we are using, every experiment is done in SST mice. So we are able to identify SST neurons. Okay, so this is the change detection task. And so why to use a visual change detection task to study predictive coding? So this task captures the core computation we care about. So the brain's ability to detect when sensory input violates its predictions. In this task, mice are presented with a continuous stream of natural images, each shown for 250 milliseconds. And most of the time, the image repeats, creating a strong expectation. So the next image will be the same as the last one. But occasionally the image changes, and the change is an unexpected event, so a violation of the brain prediction. So from a predictive coding perspective, this unexpected change should generate a prediction error signal. And this mismatch drives a cascade of activity that ultimately leads to behavior, because the mouse has to report the change by leaking to receive a reward, as we can see here that he's doing correctly. So this task gives us a clean behavioral readout that is leaks that tells us when the animal has detected a violation. And combined with our neural recording, we can map where prediction errors are encoded in the circuit and possibly how psychedelics reshape this computation. Okay, regarding the passive block, here are the details of the stimulation. I won't go through, I won't read all the details, but first we perform a receptive field mapping using Gabor stimuli presented in a 9x9 grid. And this gives us a high resolution map of where each neuron responds in the visual space. Second, we measure surround suppression. Here, instead of mapping the entire visual field, we focus only on 5 locations near the center. And this is a practical constraint because fully mapping surround suppression across all positions would just take too much time, especially given that we need to complete this protocol both before and after drug administration. So by restricting the central location where we expect the strongest responses, we hope to still characterize surround modulation decently. Okay, here we see the data set. So in panel A, we see again the structure of the session. So we have a change detection task happening before injection. So this is the pre-epoch. Then we have saline or psilocybin. And then we have again a post-injection epoch with the change detection task. In panel B, we see that we had like 10 animals, and each animal underwent 2 experimental sessions. So on day 1, it's always a saline injection. And on day 2, for 8 out of 10 animals, we have a psilocybin injection. And for the other 2, we have an additional control with another saline injection. Okay, in panel D, we see the brain rendering of all the probes used across all the sessions. So we see that we targeted mainly like visual areas, but also some cortical frontal areas. And in panel E, we see the number of cells divided by session with saline and session with psilocybin. Here we have we identified the fast spiking neurons just by looking at the waveform duration. So in average, we had around 550 cortical. So here I'm just focusing on cortical cells because this is what we have been looking at like until now. So we had 550 cells per session in average, and these are all good units that passed, like single units that passed quality metrics. And in total, almost like 10,000 cortical single units. Regarding the optotagging, here we see an example session. So the optotagging was very successful. We're very happy because we could identify many neurons and it worked in almost every session. So in panel A, you see the laser pulse. And in panel B, you see the firing rate Z scored happening during like the laser pulses. So you can see on the bottom these clearly activated cells that are really briefly active during the laser pulse. But you also see that there is a big group of neurons that are instead inhibited, that this is shown in blue. And indeed, in panel C, you see the average activity of neurons divided by the direction of the modulation. So of course, it's expected to have many inhibited neurons because, yeah, we are activating a major interneural class, somatostatin. So in average, we had around 60 neurons per session optotagged. So that corresponded to around 9% of the neurons. And if we look at the number of optotagged neurons per area, we see that they're mostly in VSP and visual areas. Okay, Here we have this is like a summary of the data that we got for each session. So on the top, you see the image presentations, color-coded by identity. In the center, you see the spikes for each area, color-coded according to the cortical area. Then we have the leaks. And on the bottom, we have the running speed in black and the pupil size in red. And now I can actually show some preliminary results regarding both behavior and neural activity. So here we have the performance on the change detection task. So we can see the reward rate over time. So in blue we see saline, and in the pink we see the psilocybin group. The black dashed line is represent the injection. So it's really striking how after psilocybin, the animal just is not able to collect any reward. You see that there is really a big drop in performance. And if we look at the different type of trials, so we see that the hit rate goes almost to 0. The miss rate also significantly increases, but also the false alarm rate goes almost to 0, with the exception of one outlier. So this means that the animal is just not even trying to report the changes. So this behavioral deficit could arise from any number of causes. It could be a motor impairment, or it could be motivation, or it could also reflect a genuine alteration in visual processing. So if we look at running, here instead we didn't see any particular change between the groups. So they're running in a similar way. And if we look at pupil size, here instead we see a significant increase in pupil size for psilocybin. Okay, we also looked first at regarding the neural activity, we looked at the general modulation of firing rate with a simple modulation index. So firing rate in the post-injection epoch minus firing rate in the pre-injection divided by the sum. And if you do this across all the neurons, so here each dot represents one session, and the mean is an average of averages. And here we notice that there is a significant difference only in regular spiking neurons. Specifically, we have a decrease in spiking. So though psilocybin may also alter like some fine-grained motor behavior, like whisking or facial movements, that running speed alone doesn't capture. So all of these can modulate visual cortex activity. So we use the face map to extract some regressor from the behavioral videos and incorporated them in a GLM to predict the activity of the neurons. So we trained the GLM on the pre-epoch and we predicted the post-epoch. And so this lets us ask after accounting for these finer motor movements, does the psilocybin effect on firing rate persists? So is the decrease we observe truly driven by the drug, or is just like simply reflect like some changes in motor behavior? And if we look at the residuals, we see that again in RS there is a difference. So they observed so there is a decrease in firing rate after psilocybin that is not explained by motor behavior. And this starts after 10 minutes after injection, and then it becomes significant after half an hour. Okay, here we see also some evoked activity. So we'll go quickly here. So we have saline on the left and psilocybin on the right. And so here 0 is the start of the image presentation. Here I'm looking at all the images that are not change images, so all the repetitions. And you can compare pre-injection here at the top with post-injection at the bottom. So in the saline case, there is nothing really going on that is really visible. But in psilocybin, you can notice that there is an increase in firing that is specific to the onset of the image and also to the offset, and somehow also in this middle area here. And we can take a look at a summary across the entire data set. So here in these heat maps, we are seeing the difference post minus pre of the peristimulus time histogram. So here black that is positive values means that in the post-injection there was more activity. And so here on the left we see regular spiking neurons, and on the right we see somatostatin neurons. And it's really striking how in somatostatin neurons, this modulation that we also saw before in just 8 neurons. In somatostatin neurons, around like 30% of the cells, they really show a strong modulation. Instead, in regular spiking, you see that it's just a small minority. So we can quantify this with an oscillation index and check this in a per area basis. And here we can notice that this is specifically, this modulation is specifically affecting visual areas, and particularly the primary visual cortex. Okay, and for the last, we also checked whether this modulation by psilocybin is dependent on the identity of the image. And we did notice that the oscillation index, so this oscillation index quantifies the modulation, and is not the same across the images. So there are some images that somehow show more modulation than others. And we also noticed that these images that show more modulation, they seem to have like long contours and very identifiable contours. So we use the contour detection algorithm to count the contours that you can see here overlaid in color and create an index. So just number of contours divided by average length. And if you correlate this index with the oscillation index, you see that there is a really big correlation. And we were, we're quite puzzled by this. We're not totally sure what this means. But yeah, there is so much to actually do with this data. So we did, of course, even more, but I didn't show it. But we are really interested in analyzing more. And if anybody is interested in collaborating or learning more about the data, please get in touch. And again, I want to thank all the OpenScope team for the great work. So.
Thank you, Roberto. It was amazing. Such an exciting set of analysis.
Thank you.
I have questions about because clearly there's interesting overlap between your talk and the previous talk, but it's also interested in, and I'm curious about what you think on global and local motion perception. What would psilocybin do in there? Because it's similar prior questions, and I think there are some work in the literature on this. So I'm just curious what you would speculate happens if you injected psilocybin in a local motion tax.
Yes, like you mean like in a surround suppression, like similar
right.
to the stimuli that were given, like shown in the previous talk.
That's right.
This sort of motion. Yeah. I mean,
right.
So first of all, yeah, we also had surround suppression stimuli because, I'm very interested in them. First of all, because somatostatin neurons are very much involved in mediating the lateral inhibition. But yeah, I mean, our protocol is kind of reduced, to properly investigate surround suppression, you should have the whole session like covered by this stimuli. So I'm not sure if we will be able to see anything there, but it's very unknown whether it would be surprising to see an effect, because I don't think anybody really showed a specific effect of psychedelics on surround suppression. I mean, there is a paper by Christopher Neal that showed an effect, but nobody did it with like neuropixels, so collecting lots of data and EFIs. And so, yeah, it's not, it's not easy to predict what would happen, I would say, yeah. But it's definitely interesting, yeah.
Any questions from the audience?
Hi, Roberto. Thanks for the talk. I'm just curious. What do you think is happening on the behavior with the psilocybin? Is the animal just not engaging, or what are your thoughts based on the data?
Yeah. yeah, it's very interesting because I mean, I was actually at first I was very scared that we would have something like this. You know, you train these mice forever, and then you inject psilocybin and they just don't do anything. So you kind of lose all the. I mean, this data is not easy to analyze the behavior because there is just a total collapse. And so, first of all, I would say that even looking at the videos, so the animals are not like totally out of it. You know, they run and they do behave. They do not lick, but if you look at the videos, you're not even able to tell whether a mouse was a psilocybin mouse or a like saline mouse. So I don't think it's an impairment on like a global like motor behavior. And my personal opinion is that it might be instead something more specific of visual processing that, I mean, in humans is very much known that after psychedelics you have like this sort of hallucinations. So my idea is that maybe their perception is totally different and they're not able, they don't understand that they are actually doing the task because they just see everything changes like constantly. This is a possibility. But yeah, it's not easy to disentangle whether it's motivation, though motivation is not known to be really affected by psilocybin too much. But yeah. But yeah, thank you for the question.
Okay. So unless Noah is available, so you can follow in the chat. They had to go to shelter. You know, they're speaking from Israel, which is under constant threat. So we prepared for this to occur, and so Javier and Federico agreed to swap talks if anything will occur about which cause this. So we're going to proceed with them, and then if Noah and Yaniv back, we'll switch to them later on. Federico?
Okay, yeah. You can hear me okay, right?
Yes, thank you. Loud and
Okay,
clear.
nice, awesome. All right, okay, so we'll get started then. So thanks so much for coming today. Today we'll be presenting our OpenScope project, which is titled From Pixels to Percepts, Understanding Texture Discrimination in the Mouse Visual Cortex. This is a collaboration, of course, with the OpenScope team at the Allen Institute, and then myself, Federico, Javier Orlandi at the University of Calgary, and James Gardner, also at the University of Calgary. So first and foremost, of course, we'd like to give a huge thanks to those at the Allen Institute that were involved in the OpenScope program and helped do the hard work of our project. Thank you, Jerome. He communicated with us so much all throughout. Alex Beat helped us a lot with the modeling, Carter with the notebook stuff and preparing our data, as well as Ahad, but I guess he's not there anymore. And then, you know, of course, Sheila, Jim, that helped a lot with the experiments. And I'm sure I'm missing a lot of people. So thank you very much. Okay, so I think we'll get started with hierarchical processing of visual information. So this was sort of already talked about in the first talk, but just in case. So the visual system is thought to be characterized by a complex hierarchy of interconnected areas that process the visual information in separate stages. And one of the sort of key ideas is that as you go down the hierarchy, you sort of see that the features that are being that are driving the responses in the neurons in these areas gets more complex as you go down the hierarchy. So for example, in V1, you might have very strong selectivity to orientation and spatial frequency. But then when you get down to, for example, the inferior temporal cortex, then you start seeing the representation of objects, for example. And this is, of course, for the ventral stream, which is different from the dorsal that was spoken to in the first project. So even though that idea has been fairly well studied, there remains sort of a question mark as to what's going on in sort of the mid-level areas. And as it turns out, it seems that there's a specific type of stimuli that might be very good at driving the responses in these mid-level areas. And these are what are called textures. And they are very simply defined as images that have statistical regularities and spatial arrangements of local features that then sort of repeat across the image, and then essentially allows you to have different kinds of images that are pixel by pixel different, but semantically, they look the same to us. And the difference, one of the very important and key aspects of these visual stimuli is that they remain parametrizable, similar to sort of gratings, Gabor filters, for example. But of course, they're a lot more complex than those kinds of early stimuli, but not so complex that you end up with objects that are much harder to parameterize. And actually, work in the non-human primates as well as humans has shown that, for example, area V2 shows a very strong selectivity to texture images compared to these noise images that we call them scrambles on our end. And these are images that have exactly the same power spectrum and average orientation, content, spatial frequency, but you've actually destroyed the correlations between these different orientations and spatial frequencies. So you essentially destroy what is the texture itself, basically. And so then, of course, there's a huge question of, can mice actually even see textures? And this is something that we ended up working, Rika and Javier and I, actually, while I was doing my PhD over there. It was my main project. And the reason we chose mice is because they actually have a hierarchy of visual areas similar to the primate visual system. But then, of course, we have all the tools at our disposal to do more in-depth circuit manipulation and circuit dissection to really try to understand what the computations are. So just some quick terminology. So we have here, I'm showing you several textures. We have what's called a family, and that's a group of textures that share a common semantic and structural characteristics governed by specific statistical properties. So for example, this looks like a thread, for example. Then over here, you have something that looks kind of like little balls or rocks. And then across the columns, what you have is an exemplar. And an exemplar is it's a unique instance within a particular texture family that is differentiated by specific realizations of the family's statistical model. And so you can see here that although these semantically look identical to me, if you really start comparing what the patterns look like, they're actually quite different. Okay, and so it turns out that mice are actually capable of detecting textures and discriminating, but I'm only going to be talking about the detection part for here. And so this is the previous work that I was talking to you about. And so essentially what we did is we had a go, no-go task for the mice, and it was very straightforward. So the mice were head fixed, and they were being shown stimuli in front of a screen. And if there was a texture, they were asked to rotate a wheel to then get a water reward. And if it was a scramble, then they were supposed to withhold movement. And again, I just want to emphasize again that the difference between these two images is the higher order statistics that actually typify a texture. And between these two, the statistics that are the same would be the low order ones. So for example, the mean luminance, the RMS contrast, and then the average spatial frequency and orientation content. And so when we evaluated the mice on 4 different families, so we actually see that they are able to discriminate between these two across all the families. And what was interesting is that we saw that there was one particular family that's had a significantly different performance as measured by D prime, the discriminability index, compared to the other 3 families. And so this actually became important later on in our work. And so with this, now the question becomes is, well, we know they seem to be able to discriminate textures from the scramble control images. So that means that there must be some brain areas that are responding to these textures. And so now we did some imaging experiments, but the caveat is that they were fully passive. And what I mean by that is that the mice were not doing any tasks. They were never trained on anything to do with texture discrimination, and they were simply we did the surgery to get all these visual areas. We injected virally expressed JGCAM7F, and then we placed them after getting the retinotopy, of course. Then we placed them under the same rig, and we just showed them different kinds of textures, 4 different families with their corresponding scrambles as well. And so I'll keep it very short. For the Whitefield stuff. So what we essentially find is that V1 and LM both respond differentially to the textures. So the texture seems to elicit a strong response. But then when you go down to LM, it seems like the response to the scramble images, which are again the noise control images, has a lower response. And as a result, the difference between these two is statistically significant. And so of all the visual areas that we imaged that I showed earlier, V1 and LM showed the strongest responses to the textures. And so then, of course, this drives the question of like, well, what is actually happening at the single cell level? And so to answer the question, we essentially repeated the same kind of experiment, but now we focused on 2 areas. So we focused on V1 and then we focused on LM, and we did 2 photon imaging. And it's basically the same experiment as in the Whitefield, fully passive. There is no behavior again. And unfortunately, due to the setup that we actually had to record the sessions for V1 and LM separately from one another at different days, for example. And yeah, so it's essentially the same sort of experiment. And so one of the things that we were able to find was mixed selectivity. So we find that there's some neurons that seem to respond to textures. Actually, the majority of them show this sort of differential response. And essentially in LM, there's many more of these cells, and the difference is actually also stronger, just like we saw in LM. But at the single cell level, we can actually even find cells that prefer the scrambles, which again, I actually have different exemplars of each scramble. So the fact that there is some sort of actual selectivity to it, it's maybe interesting. And now I'm going to skip ahead from what is on the paper. I'll show you about the paper later, just due to time. So now I will show you the sort of the main finding that from our paper that we were hoping to sort of improve upon with the OpenScope. So if you recall, and when I showed the slide on behavior, we saw that there was this family that had the worst performance. It was the ROX family. The mice were discriminating it between their scramble, but they had the lowest performance compared to the other 3. And what we can do is, since I said that the textures are parameterizable, we can actually measure in this statistical space the distance between a texture and another texture, but in this case, a texture with its corresponding scramble. And we measure these distances. And what we see is that there seems to be a correlation or a relationship that when you see the behavioral D prime, the family again that has the lowest performance was the one that had the closest distance in this image statistic space. And then now we looked at the passive 2-photon experiments, and what we did is we trained a classifier to tell me when a particular brain activity belongs to a texture or when it belongs to a scramble, essentially repeating what I'm doing on the behavior side, but on mice that were, again, not trained to do this, and we're simply just observing the textures in an awake mode. And again, we find similar relationships. And the difference between the ones that don't have a strong color, like the less opacity ones, is these are the ones from V1, and then the ones that are solid in color is the classifier performance that comes from LM. And again, we sort of see this similar relationship. And then we also have, of course, the opposite. So yeah, neural accuracy and behavior. And I'm skipping a lot of the paper, but if you want to learn more about this, this is the paper that sort of where everything that I just talked about comes from. So now, of course, these leaves a lot of unanswered questions. So 4 families is definitely not enough. And so the linking framework, so what I just talked about, can the predictability be generalized across a larger set of texture families? And that's like one of the unanswered questions. We have another one. So all the imaging that we did was fully done passively. So now there's an interesting question of like, well, if you do actual behavioral training on this and you actually image these mice, do you see that the perceptual training will shapes the texture representation in some way? And then finally, so due to the way we had our setup, we could only do either V1 or LM separately. So then the question becomes, is the textual representation more separable across the dorsal stream now that we have data that is, acquired simultaneously, allowing us to very nicely probe at the neural subspace between the different areas that we recorded. And so then with that, I can start talking about the OpenScope experimental pipeline. So we chose only the SLC 17A7 mice that express GCAM on this with this promoter. And we then, after they do the surgery to implant the window, then we did retinotopic mapping. Following retinotopic mapping, we then now separate the mice into 2 groups. We have the passive and we have the active group. So the passive group, what we did with these mice was simply image them in a similar manner as we did in our earlier work, in the earlier mouse work. So they didn't get trained on any sort of behavior. They were simply habituated to imaging, and then we showed them the images that we prepared for them, which is now a lot more. I'll talk about the stimuli a bit later. Then the second group is the active group. And so these mice would go on behavioral training where the first phase is exactly the same as the Allen Institutes with just gradings. And then rather than switching to like natural scenes, we then actually switch to a single exemplar per family. I'll again talk a bit more about this task in detail. But once they're trained, we then move them onto the OFIS setup. So we would do 2-photon imaging while they are doing behavior. And then at some point during these imaging plus behavior sessions, we switch to a different protocol that is more complex and is supposed to show good generalization for the mice. And then at the very end, we did passive 2-photon imaging of these mice once they were done training. So now let me talk very quickly about the passive stimuli. So we have 16 texture families. Each one has 28 exemplars. And then in total, that means we have 448 unique images. Here are all the families. And then the passive imaging experiment details. So each of the 448 unique images was shown 10 times, so 10 repeats. And the experiment is basically the same as the ones from the Allen Brain Observatory. So 250 milliseconds of stimuli with 500 milliseconds of gray screen. Then the order of the images is fully randomized. And then the active group has some passive sessions at the end that are exactly the same as this. So now I will quickly go over the go, no-go tasks training. So like I said, phase 1 was purely gradings. Phase 2 is 8 families with 1 exemplar each. And then the way it works is you have the textures. And then in this case, now I'm saying that this is the same family. And then at some point, it switches to either a different family or it stays on the same. And if it switches to a different family, the mice are supposed to respond by licking a spout, and we detect that as the response. Then phase 3 is exactly the same 8 families, but now there's 28 exemplars that consist of XY shifts from a reference image that we started with. And the way that we did the experiment was such that once the mice were proficient in phase 2, they were moved on to the OFIS setup. And once the phase 2 performance returned to baseline, then we switched them onto the phase 3, allowing us to capture the moment where they understand the new rules of the task and thus show generalization across exemplars. So then here I can show you very quickly a diagram that shows phase 2. So phase 2, it's exactly the same image across. At some point, it switched to a different family. But then phase 3, you have an exemplar 4 and then exemplar 9, which is exactly the same image, but it has now been shifted some amount. And then at some point, it switches to a family, and that's when the mice are supposed to respond. So the mice are supposed to learn to ignore the changes from going from exemplar 4 to exemplar 9. And so active stimuli detailed. So phase 3 is the 28 exemplars from 8 different families. And we actually had 2 levels of difficulties. So we have small versus large shifts. In the interest of time, I'm not going to talk too much about it, but basically you can sort of see here the distribution of shifts that we chose. Okay, and so data generation plan. So we, like I said, there's the passive group, there's the active group. And then when we were doing imaging, the most important thing is that we recorded from 4 different brain areas this time. And we actually did 2 depths. So we did both layer 2, 3 and layer 5. And in addition to that, we to understand sort of the effect of time as well in these representations, we asked the Allen Institute team to give us consecutive sessions separated by a bit of time. And each time that they would do a session, we would ask them to match the same cells across different sessions. And then here's a graphic that shows the different animal sessions, and then colored in like blue, orange, and green, we have whether it's a fully passive animal, whether it's the active with a single exemplar, phase 1, or if it's the phase 3 where there's like multiple exemplars per family, with the different shifts. And then at the very end, the mice have their passive recordings. Okay, and so with that, I will pass it on to James for the talking about the results.
I have a little question for you, Federico, as he's setting up.
Yeah, sure.
So the other projects do not use textures, and so I'm just curious because you spend a lot of time thinking about efficient coding and all of that aspects. Do you think that framework of thinking also apply to gradings, or do you think there's a specialization?
I mean, in theory, it should apply because to me, the idea of efficient coding is just that the statistics of the input images are just as well represented in the neural representations as they are when we generated the images. So I assume that gradings wouldn't be anything more special, and they should follow the same sort of rules. Now, I mean, admittedly, gradings are definitely not not a naturalistic stimulus. So I wonder if there is something there, because I think anecdotally I've heard that if you look at the representation of vertical and horizontal objects in societies that are more what's the word, like like tribes in Africa, I think was what they were showing. They showed that they didn't have as strong of representation of horizontal and vertical orientations in their brains, because they don't live in a place like, for example, us, where there's just buildings everywhere, that there's just straight and horizontal lines everywhere, tables, monitors, everything. And so I think that might also play a role into that.
Yeah, the history. That's right.
Mm-hmm.
I had another question. So
yes,
when you decided on the textures, did you kind of just pick what you thought was like clearly discriminable textures, or like, what was your hypothesis for how, what parametric properties of the textures would actually be important for discriminability? Like, did you do something systematic to determine what properties you wanted to use?
Yeah, so I let me, while we're waiting for it, then I have a slide that then I didn't. Okay, so the way that we picked the texture families was in a way that tried to have some groups of families where the statistics are very similar from one another. And so here I'm actually skipping a lot of I guess a lot of the prior thing on what I'm talking about image statistics. But if you want to learn more, like it's on the paper that I shared earlier. But it's basically a model that has handcrafted statistics that get extracted for each image. And then this model actually ends up being used to also synthesize textures by Javier Portilla and Eero Simoncelli. This is from the year 2000. And so essentially we use that model to then pick our textures because we wanted to have some textures that in different kinds of statistics are close to one another, so that we can really try to separate hopefully the specific group of statistics that are being extracted by the mouse. So by the mouse visual cortex. So we have basically 4 groups. So the energy statistics, the linear statistics, the so-called spectral statistics, and then the marginal, which are actually this time we forced them to be identical. So that's why you can't really discriminate different families from one another. But that is basically the sort of a heuristic that we use to pick the families. Because the idea is that, well, we think that it's the energy, the one that seems to be driving the if you recall, like the linking framework where the rocks one has the smallest amount of distance. So that relationship only held true for the energy and didn't really hold true for the linear and spectral statistics. So we were hoping to find ones that are really, really quite different from each other, so that we can really confidently say, like, hey, it is the energy. Yeah, thanks for the question. I don't know who asked it because I guess I couldn't see the camera.
What's Carter?
Oh, Carter. Okay, thanks, Carter.
So just a little bit of a recap on what Federico was talking about with the sort of paradigm setup for the different data that we've collected from this from the island so far. So first we have our passive sessions. These, again, no behavioral data was collected during these, and during these the animals were presented with 16 different families of texture images, with each having 28 different exemplars, and this is to sort of get the innate response to these different visuals. During these, we collected all sorts of different calcium data from the areas mentioned, the 8 different areas, 4 different regions at 2 different depths of the visual cortex. For example, we have ROIs, the mask of these ROIs, identifying the different cells, as well as some of the neural responses to the different visual images or to the different textures set up. So here for DFF, for example, from our layers 2, 3 of our V1. We also have our passive sessions, or actually active sessions. In these, we have different stages, as Federico mentioned, but just to sort of recap, we have an initial stage, which is a simpler stage with 8 different families, each only having 1 individual exemplar, followed by a similar setup, but now with 28 exemplars, where an animal has to lick to be able to identify the transition between one family and another, to sort of see if they are able to discriminate, using a broader set of exemplars to see if they're able to extrapolate those beyond. This is followed up by a passive viewing session to see sort of, again, what the response is after they are no longer naive, after they've gone through this sort of training or sort of active paradigm. So during this, there's the simultaneous recording of all of the neural data that I talk about, as well as behavioral data. So this will give us our lick response, so telling us the animal's ability to discriminate between these different textures, as well as things like reward volume, running speed, things like that. From just some sort of initial behavioral analysis using a D-prime discriminability measure, we're able to see that the mice in this case, are able to actually distinguish between the different texture families. This is consistent across both stages of our active session, with the 1 exemplar and 28 exemplars both, showing that they are able to at least behaviorally generalize to a wider set of textures, being able to distinguish these texture families apart from each other. Looking at the cellular response, we looked to see whether individual cells were responsive to specific images or specific families of images. This here is just a scatterplot raster of the cell responses, so a cell either being active or non-active, here having a D prime of above 0.5, with the red lines indicating families of different images. This is some examples of data from our passive sessions, where we see a little distinction. We see some cells that are nearly universally responsive to just texture images, but not necessarily selective to any specific subset of those. Here we see during the active sessions, we see a little bit more structure going into the actual image families themselves, seeing some cells that seem to be responsive to subsets of families, indicating that there's some sort of information about these families being carried within these individual cells. And again, indicative of some sort of discriminability between them during these active sessions here. Just some data from both the V1 and LM, some areas that Federico mentioned were seemed to be important for texture processing in his previous work. We also see the cellular response for the passive sessions, or the passive part of the active session. So animals that are no longer naive had gone through their active sessions and are now looking at passive viewing, where we do not see some of that structure that we saw within the active persist. This is backed up by looking at the proportion of cells that responded significantly to these different that responded significantly to one texture family specifically, or a subset of texture families specifically, looking in the proportion of cells in the passive sessions near baseline, whereas in the active session we see a significant increase. And with specifically looking at the V1 as well as layers 5 of the LM and AL seem to have a particularly higher proportion of these cells, indicating that some of the information may be carried in these regions. Looking at the passive sessions afterwards, we see sort of a similar story where we see these active, during the active session, this high proportion going down significantly afterwards, dropping even after the training sessions. We then also trained some model to be able to see whether we are able to distinguish between the different families based off the neural data, specifically using sport vector machine to look at some, to see if there are any nonlinear dependencies, using the delta f over f prior and post the image presentation. What we see, at least for the passive session, is that these animals, the neural discriminability seems to be near baseline for all the different cortical regions, for models trained on all the different cortical regions across basically all of the sessions. So as time goes forward, there does not seem to be an increase in the neural discriminability. This is not the case for the active, where here's an example for a model trained on our V1 looking at, you can see here the first two sessions, or the first sessions being our active sessions with only one exemplar, then our active sessions with multiple exemplars, seeing that we're able to generalize, the model is at least able to generalize beyond to this higher number of exemplars. Whereas the passive sessions after those active sessions, it seems to collapse to near baseline, with the model not being able to pick up on it, or having, again, baseline accuracy on the its predictability in terms of identifying the texture family based off of the calcium data. This is somewhat consistent across the different regions looked at. So here we have, again, the all 8 regions, V1, LM, RL, and AL, layers 2, 3, and 5. And again, lining up with the proportion of cells that seem to be selective to these different regions, or selective to the different families, we see that the V1 is with layer 5 of the LM and layer 5 of the AL seem to be performing particularly well. Specifically, we see maybe a little bit of a drop in the first sessions of the generalized experiment, where we do 28 exemplars, but this seems to recover and then again collapse when we get to the final passive sessions. Another interesting thing about some of the data that we have, again, with the ROIs and with the cell masks, is that we're able to track cells across different sessions. So Federico mentioned looking at cells across different days. By doing some sort of affine transformations, we're able to match cells from one region of the cortex to another day after day. And we then did try to see how these internal representations shift. So this is sort of an interesting thing we can do with some of this data. We trained the model, it's the same sort of support vector machine model on a one session, and then tested on the subsequent session using the cells that we were able to match, matching about on average about 65% of neurons between different sessions. What you can see is that there may be, again, this is just a specific example from one animal, but that there is a drop between our sessions with 1 exemplar to 28 exemplars, but this seems to increase before, again, collapsing at the final passive viewing with the 16 families. So just to summarize some of the data that we have, that it's a large data set recording from multiple different visual areas, cortical depths, sort of expanding on the work that Federico did, using these texture images, which again, although they're complex, are very easily parameterized to be able to quantify how the different aspects of these textures impact or how they are encoded within neural data. It also combines both neural and behavioral data being recorded simultaneously rather than being recorded separately, being able to see links between behaved or having a stronger link between behavioral and neural tasks, as well as it allows us to explore the internal representations of these visual textures and how they are used in perceptual decision making. Thank you.
Really impressive first results. The contrast is striking between the active and passive context. Do you think that's a deployment of attentions, or it's really a development of the features through training? What's your opinion?
Well, again, that is something that I think about. I've been thinking about quite a bit. I'm struggling to think of something else apart from attention that would be the case, just because there is such a striking difference, I would believe, between some of the stuff that we have here and Federico's work. There's a whole bunch of different methods that I've used, and they all sort of seem to point towards the same end result. So I think that I think it may be just a function of the fact that we have such different paradigms between our final active sessions and our passive sessions, that 16 different families, doubling the number of images that we essentially have. It may be difficult for the animals to generalize whatever they've sort of created within those tasks or within those active sessions to those 16 different families. But even looking at just the sort of training those models, being able to identify just the same families that are in both, for example. I've done both where you've trained it looking at all of the families that are all 16 families, as well as just the 8 that were in the previous, and you still get somewhat similar results in both of those. So I would guess it's something to do with the attention of the animal, rather than some sort of actual setup of the experiment. But that's something I'm still trying to grapple with.
There is actually
Okay.
one other big difference from the earlier work. So when I did the experiments for my PhD work, the mice were positioned such that they were facing the monitor straight on, and I was showing the stimuli in their binocular zone. So I have no clue if that could somehow affect their attention mechanisms or what it could be, because the OpenScope experiments, on the other hand, they were like monocular, full field, which was different. So maybe that could also play a role in it, but I'm not too sure.
And for sure, you have more analysis to do on this front.
Yes.
But any questions in the audience for the texture team? Okay. I mean, we're back at 10, so let's start with let's move on with Noah. Thank you, Javier, James, and Federico, for this great presentation. And first, shout out to the Israeli team to marching on with science despite the adversities.
Thank you very much, Jerome. Noah, you here?
So, hi everyone, my name is Noah. I'm a student in University's lab at Weizmann Institute, and I want to thank you very much for this opportunity. I will present our OpenScope project that I do with close collaboration with Daniel Ditch and Alon Rubin. In this project, we aim to examine the effect of prior experience on naturalistic stimulus presentations. One of the biggest questions in neuroscience is, how does the brain make sense of real-life visual scenes? But real visual input is incredibly complex. So in the lab, we usually take a reductionistic approach and show synthetic images that are easy to control, but rarely reflect the richness of real-life visual scenes. This focus on synthetic stimuli stemmed from the limitation of technology that was available at the time, single cells recording, and this ability to identify stereotypic responses of single cells to synthetic stimuli led Hubble and Wiesel to the discovery of orientation tuning in the primary visual cortex, and to the establishment of visual system hierarchy. These findings serve as a very useful theoretical framework for understanding the visual system processing, mainly as a feed-forward hierarchical system. But as the visual research progressed, this framework did not fit newer findings, such as processing of complex stimuli and feedback communication. And today, with technology advancement, we can record large population of cells. And this population recording uncovered very important insights about population coding that could not be found exclusively from single cells activity, such as the case you see here of spatial navigation in rodents. Combining large population recording with complex stimuli into today's visual research has raised an opportunity to discover how visual system processes natural stimuli beyond traditional models. For example, by using large population recording, it was possible to look at the stability of neuronal code in response to a complex stimulus in the visual system. In this work, you see an example for the activity of a single neuron in V1 in response to a simple bar at different orientations. The tuning of this neuron to a specific orientation is indicated here in high activity, marked in yellow. This neuron exhibits relatively stable response across all 56 trials, as you see here. But when recording this neuron's response to a movie, this neuron's response gradually changed across trials, what we call representational drift. So while the representation of a simple stimulus can be stable, it was found that for response to a complex stimuli like a movie, the responses drifted. And similarly, in the work from our lab, analyzing Allen Brain Observatory dataset, the same phenomenon of representational drift of the visual cortex was also shown in higher visual areas and also occurred in shorter timescales from minutes to hours. The use of large population recording with complex stimuli revealed something interesting. Although individual neurons' response to movies drift over time, the internal structure of the population activity remains highly stable. In the same work from our lab, here you can see the response of approximately 400 neurons to natural movies, and they are sorted by their peak activity. To better understand the structure of this population response, we represent the activity at each time being of the movie in low-dimensional space using the dimensionality reduction algorithm. Each dot corresponds to the population activity pattern at a single time point in the movie, as you can see here. In this visualization, points that are closer together represent time points with more similar population activity patterns. So when we color this low-dimensional space according to time in the movie, from blue at the beginning to red at the end, you see that nearby points in the movie are also nearby in the neuronal representational space. This created a ring-like structure, and very importantly, this structure remains stable over time, even though the activity of the individual neurons seems to drift. And while this original project of Daniel was focused on the code stability of the visual cortex, visualizing this activity in low-dimensional space revealed something very unexpected. Daniel noticed that a striking similarity between the neuronal representation of the beginning and end of the movie. You see it here. The blue and the red are close to each other. And this similarity is not trivial because those two frames are visually distinct, as you can see here, and also here in the visual stimulus space. Since this movie was shown in a loop, the interesting finding raised the question of whether these associations are formed due to their temporal proximity. Because the movie was presented in the loop, each movie was followed by the next one, so these two frames, the last one of the previous one and the first one of the other one, are always temporally close to one another. So this is what we wanted to test. And this led us to address two main questions in our research. One, if could distinct visual scenes be associated through their learned sequential order and temporal proximity? And if so, can this persistence of the association over time constrain visual system flexibility? And to address the question, we proposed this experimental design you see here. The full experiment lasted 2 hours. The first half was dedicated to answer question number 1, and the second half to question number 2. The mice were recorded using neuropixel probes. Each animal had 6 probes, which recorded from various visual areas, but as well as thalamic and hippocampal and midbrain areas. The experiment was conducted with 2 groups of mice, as you see here, 7 mice in each group. The group A was usually the experimental group, and group B served as a control. Now we will go deeper into each of the aims. So the first half of the experiment aimed to determine whether the visual system can form associations between visually distinct natural scenes based on their temporal proximity. So to test this, we familiarized each group of mice with the movie presented either in a loop or in a forward-reverse manner. So the first and the last scene are either temporally close, like in the previous experiment, or temporally separated, like you see here. This dissociation enables us to see if the temporal proximity is what causes this neuronal association that we see in the ring. And this experiment is counterbalanced, so both groups went through 2 blocks and experienced both conditions. We hypothesized the following. For group A, which was familiarized with the loop condition first, where the first and last frames are temporally close, we expect a ring structure to be formed like in the previous dataset. And in the second block of group A, the temporal relationship between the 2 frames is now broken, which is now the forward-reversed condition. And if the association between these 2 frames stem from associative memory in the visual system, this association might persist also in this condition when those 2 frames are no longer temporally close to one another. So we expect the ring structure to remain. And this can also be quantified by calculating the similarity between the activity patterns of the first and last frames. And we expect the similarity to be high in both blocks, either in the loop or alternating conditions. This represents that the associations we see in the ring structure also can be calculated through similarity of the population itself. For group B, what we expected, they were familiarized with the alternating condition first. So the 2 frames are temporally separated, and we don't expect this ring structure to form if we expect that what causes this ring is actually the temporal proximity. But in the second block for this group, I do expect to see this kind of formation because now they are viewing these 2 frames temporally close. And also, when we calculate the similarity, we expect to see low similarity in the first block and then high similarity in the second block for group B. We're currently in the process of analyzing the data, so we still don't have an answer to this hypothesis specifically, but we will show some interesting preliminary results from the dataset. Okay, so you can see here neurons. They are sorted by their peak activity. There are neurons from V1. And this is sorted by the activity in the loop trials when they view the movie in a loop, in a forward. And what we expected, it's what we got. We see that we identified a ring structure in the reduced neuronal space, also in this dataset. And you can see that the first and last frame, which is the blue and the red, are also close in this low-dimensional space. Sorting the same neurons activity by reverse presentation now actually showed something interesting, that many neurons actually show some kind of stability which means they are firing in response to the same stimulus even when the movie was actually presented in reverse. So you can see here in this distorted flip dark diagonal of the peak activity. But while some neurons exhibited stable tuning to reverse presentation, some neurons actually showed change in tuning. And this plot, you can see two neurons, for example. You can see here the different phases of the experiment. You can see the first forward in a loop. This is how we call it. And then there is the forward presentation and the reverse. This is the forward reversed condition. I divided it by the direction that the movie was presented, but this is actually was shown interleaved. And you can see here there is this neuron that is actually showing stable tuning throughout the experiment. I actually also flipped the reversed to be actually similar to the same stimulus as the forward presentation. So it will be more clear if the neuron is actually stable to the stimulus or the time in the movie. So here you can see this neuron in particular is actually stable for the same stimulus in the movie throughout the experiment. But we also see cells that actually change their tuning for the reversed presentation of the movie. As you can see here, this neuron is showing a very stable tuning to the forward presentation, but then when the movie is presented in reverse, it actually changes its tuning. So these changes are very interesting and maybe can reveal some non-trivial coding of these neurons. Okay, so in AM1, we used large population recording combined with complex stimuli, and it was necessary to identify a ring structure with an unusual neuronal associations. But even in modern visual experiments using complex stimuli, it is common to repeat the exact same stimulus over and over again. However, real-life visual scenes are dynamic and constantly changing, like this face example you see here, even though our perception usually feels stable and coherent. And this raises an important question of how does the brain adapt its representation of a stimulus when it changes gradually compared to when it changes abruptly. And this inspired us to test this with the following experimental design. You can see here that we also have two groups. Both of them had the baseline phase in which they saw the same movie again and again repeatedly for 20 repeats. And then each of them went through a different phase. Group A went through a morphing phase in which they saw the first version of the movie, like in the baseline, but now the movie is gradually changing through 50 repeats until it becomes something else in the last version. Group B viewed only the last version repeatedly for 50 trials, so they actually experienced an abrupt change in the stimulus. And then both groups went through a recall phase in which they saw first version and last version interleaved. So for this experiment, we designed a novel gradually changing movie. In every trial, the movie is presented with a slight change. The beginning and end remain the same, while the middle scene changes gradually across trials until it becomes something entirely different. You can see it here that these two scenes actually remain the same throughout the experiment, but when you view this frame that is representative for the middle scene that is changing, you can see that it begins as this frame, but then it changes to something entirely different. And now you can actually view it. We are showing you the first and last version of the movies. You can see here that this one is the original and this was totally morphed. There are also stationary scenes that look identical, but also a scene that is changing. Our scene were visually distinct from each other with objects in different sizes, moving in different directions and orientation. And we try to maximize the difference in representation between those scenes. And the changes between the versions were present in the entire screen. We made sure of that. So after establishing the gradually changing stimulus, we can now look at the activity of single neurons. You can see here one cell, for example. Each row is the activity of this particular trial. You can see here there is the baseline phase and then the morphing, and then the recall phase, which I ordered it by the stimulus. So there is the first version, the M1, and the M50, the last version. And you can see here this particular neuron is actually tuned to the first scene of the movie, which is stationary. So it makes sense. This scene does not change throughout the experiment. So we actually see cells like this in both groups, where in the stationary scenes, they just remain stable throughout the experiment. But then we see something very interesting in the morphing phase, that in group B, in which they saw the change abruptly, we see that cells that have some sort of tuning to this particular scene then just change their tuning but remain stable throughout this phase. And we see cells, of course, like this in group B. But then in group A, when they had a gradually changing movie, we actually see very interesting patterns. Either the cells were gradually changing or changed their tuning abruptly. So this is two types of patterns we see, but we actually see many different, very interesting patterns of changes in of the activity of the neurons. That is very significant, can be seen significantly because of this gradually changing movie. And just like the initial example of gradually changing faces, we wanted to know whether the process of gradual change in the stimulus can constrain the degree of change in the stimulus representation. So to test this effect, we focused only on the changing scene now and correlated the average population activity per trial in the morphing phase with the population activity in the baseline phase for both of the groups. And what we expected is that the experience of morphing of group A will restrict the degree to which the representation will change, meaning that it will always be more highly correlated or more similar to the first version in the baseline phase than group B. As you see here, the correlations are higher. But if the morphing has no effect on the change in representation, we expect that the correlation of group A to just go as low as group B. And this is also an ongoing analysis, but we want to give you a glimpse of this data set. And you can see here also one cell, for example, and you can see that using a gradually changing complex stimulus can reveal a very interesting changes in the tuning of single neurons. And this might uncover a phenomenon that might not be explained just by the presenting the same stimulus over and over again. And looking at this specific neuron's activity, we see some interesting changes, like this one in particular. To further investigate this change in the tuning of the cell, we can examine how the stimulus changed in those trials and try to detect what this neuron is sensitive to in the change of the complex stimulus. And this kind of effect can also be differentiated between lower and higher visual areas, as they have different receptive fields properties, and it is recorded in this experiment. So to summarize, we initially hypothesized that distinct visual scenes can be associated through their learned sequential order and temporal proximity, and the persistence of this association over time can constrain visual system flexibility. And to address this aim, we created an experimental design with a dissociated relationship between frame identity and temporal proximity and a gradually changing movie. And I want to thank, of course, all Allen Institute and specific Jerome and all of OpenScope team who were very kind to us throughout this experience and gave us this very amazing data set that we're feeling very grateful for. And I want to, of course, thank Yaniv, the PI in my lab, and to Daniel Ditch and Alon Rubin, who is working very closely with me on this project. And thank you, of course, for listening.
Thank you for this wonderful presentation. I wonder if you could speak a little bit. So in the, because I think you've been focusing on pyramidal cells, and do you anticipate the inhibitory network to sort of play a role in this gradual change? Because you see many of our projects are thinking about this. What's your thinking?
I actually, we don't really have the identity of the neurons. I mean, we cannot identify explicitly which neurons are interneurons and which are not. I did see in this data set neurons that have some properties that look like interneurons, and I could see some very interesting patterns, but we did not look further into it and try to really classify the neurons by their identity, but it can be very interesting, and I do look forward to further investigate it.
That's right. That's right. Any questions in the audience for Javier?
Yeah, hi, Noah, very interesting work. I'm just curious because these movies seem to have a lot of detail. How did you choose what movies to use as the visual stimulus here?
Yeah, that's a really good question. We did something novel because we wanted something that is gradually changing throughout the experiment through 50 repeats, and it will change entirely and also include stationary and changing scenes in the movie. So it made us use a novel movie and to create it ourselves. I think the important thing is to create something gradual and that it's also not focused in one specific location of the screen, that it will change gradually on the entire screen, and using different objects on the screen that's moving in different directions and in sizes, so it will not be very specific, and to really create something that is complex. I think this was the main idea, not to create something that's very simple. I hope it answered the question.
Perfect, thank you.
Is
You
it
might
okay?
not
Like
if
I can
they're-
add a little bit
Oh,
because
please.
I was the one like that actually took the mission to do it, and like if you're looking for something, like if we chose specific features that we know that are relevant to the visual cortex, this was not done. Like the whole point was to first create free scenes that will have some naturalistic statistics, not just random stimuli, and that the scenes will be different enough that the visual cortex will fire differently for different times in the movie, because we are going to change only the middle part. If there is some similarity between scenes, this might be problematic. So we try to have like the first scene will have horizontal lines. The middle scene will have like vertical ones, and the last one will be more like, you know, circles and black, but it wasn't like very methodical and really in like calculating the power or stuff like that. And in terms of the change, like we changed the transparency of, like we had two scenes, like the first one and last one, and each object gradually changes transparency by 2%, and some change their size by 2%, and it's happened to the entire screen. But we don't have control about what's the statistics that happen like in each version, and you might have different objects that create something new. We didn't control for that at all. But like the point was that maybe the gradual change will enable us to pinpoint, like based on neuronal activity, like something changing the neuronal activity. Now we can try to look at the stimulus and understand. Hope it was clear. And
I feel this was helpful to provide context. Then the movie is, yeah, everybody will have access to it. You could do some more higher order analysis like you would do, Javier, with textures.
We made sure that just pairwise statistics, like pairwise similarities in the versions gradually change, and there is no abrupt between consecutive versions and stuff like that, but nothing very, very methodical.
Yeah, and I think it's very good actually that you have very distinct features between the different movies, and then you can, yeah, fine-tune the results based on the movies. Yeah.
Okay. Any other questions for the group? Well, thanks so much for this amazing presentation. And please feel free to reach out to the team's advisement if you want to collaborate with them. The next presentation is from Carter, who would introduce a number of notebooks to help navigate this dataset with code. Carter, please introduce yourself and then
Hey everybody,
the
I'm
stage.
Carter. You guys hear me? Okay, and let me share my screen here. So I work on the OpenScope team with Jerome, and I collaborate with the external teams. So in addition to helping package their data and actually push it and releasing it on Dandy, I've also worked on the OpenScope databook. But before I talk about the databook, I want to just briefly talk about Dandy. We plan to release the data sets for all of these external teams that have been presented in the next several months, and they will be delivered and released on Dandy. So if you aren't familiar, you can just go here and you could just search up OpenScope, and then these should be publicly and freely available for you. So just to give you a little tour here, we have our previously released data sets from last year, for instance. And you can go here and we have a lot of sort of metadata, authorship information, funding information. And if you click on files, you can actually see the files themselves. And the files are in NWB format. And it's actually non-trivial to get the NWBs packaged and pushed onto Dandy like this. We spent a lot of time, heavily considering how to make the data as usable as possible. And so that's a lot of what I've done along with Ahad Bawani and some other people. And that was just basically primarily an effort to help, push our data in a way that's actually highly usable, highly reusable. But it doesn't completely solve the issue. So that's it. To get into the data book, that was sort of the next step. So we put a lot of work. Well, I'll just here. Can you guys see this? Down here.
Yes.
Okay. So this is Dandy. And the next step after we sort of worked on getting our data actually pushed and packaged was the OpenScope data book. So NWB files, when we started OpenScope project, NWB files were very new, and I think they're really unfamiliar to people. And also there's just a lot of content in there, and it's not immediately obvious how to access all of the data. In addition to that, we have our two primary modalities. We had our two photon mesoscope data, and we also had our electrophysiology neuropixels. And there's all these common kinds of analyses that existed at the time, and people at the Institute and elsewhere were sort of just reinventing the wheel and reinventing a lot of code to try to just do a lot of these basic analyses on all these new data sets. And so we wanted to address a couple of concerns in one with the OpenScope data book. The first one is what I talked about, familiarizing people with NWB. The next is basically creating a repository of code so that we can actually quickly run these analyses without doing a ton of work to fit to specific data set, just sort of those secondary technical details that kind of inhibit progress into investigating the data. And we wanted it to be in one spot where you could actually do it quickly and easily. So these were the sort of goals we set out to address with the data book. And we decided to do that with a Jupyter book. And so that's basically just a website, a web page, organized set of Jupyter notebooks. And we host it on GitHub Pages for free, openly available. And importantly, though, the notebooks aren't just static documents. We actually also provide the ability to launch the notebooks onto compute platforms. So this is, I think, really important for getting reproducibility, because anybody who's actually tried to use someone else's data set that isn't theirs knows that there's a lot of bespoke information that goes into actually like digging into the data, right? You spend a ton of time trying to set up the appropriate environment. Usually you have to know a lot of weird specific details about the data set, weird exceptions and things like that. And this is just a huge barrier to entry for reproducibility, right? And Jerome at the beginning talked about how OpenScope's whole mission is about sort of opening our resources to the world and our data to the world to kind of create a collaborative ecosystem. So the OpenScope Databook is just one additional pillar in that ecosystem, so that people can quickly jump into the data like I already mentioned. So it's easier just to show you guys the data book rather than talk about it with bullet points. So you can see it here on GitHub. We have a README, we have our environment. This is a little table of contents, and people can, write their issues. We wanted to utilize as much, sort of existing infrastructure as possible. Once again, to make the ecosystem, we didn't want to reinvent the wheel, and we wanted to make the ecosystem very familiar to people. So we also allow for pull requests, people to contribute, and anybody who contributes gets added to the authors list of the data book. You can actually see the website here. This is the authors list, and it gets automatically updated with people's commits. And you can see all the notebooks that are consistent of here. And like I said, this is dynamic. We're updating, we're adding new notebooks. So people have new analyses, new libraries, powerful tools that help facilitate analyzing NWB files. I'd encourage you to contribute, and we're sort of always updating and maintaining, accepting ways to grow and improve this tool. So to go over kind of what actually is in the data book, we have sort of this section, which kind of just goes over how to actually use an NWB file and retrieve them from Dandy. So if you don't want to actually go through the Dandy archive GUI, you can sort of type in the important information here and actually download an NWB file. This is one from, I believe, our visual coding data set. You can see we have behavioral information like the wheel. We have eye tracking information. This one has opto tagging. And then we actually have, the electrophysiology here, which is stored in the table. And then, finally, we also have the stimulus information. So I believe pretty much all of our NWB files have those key elements. So it has the behavior in the form of running and eye tracking. It has the actual, recording, either 2 photon or neuropixels, and then the stimulus information, along with, some additional metadata like the file create date and stuff. So we have our visualizing NWB files. Once again, this is actually sort of digging into what's unique about our data sets, not just NWB files in general. And it goes through the 2 main modalities. We have notebooks to describe quality metrics and get insight into how we're doing our internal processing and evaluation of our data. LFP, let's see. Yeah, and then sort of some basic analyses here. We have more first order analyses. I won't go into all of these, but just to highlight some important ones. We have this one to describe how we identify opto tag units, and there's not really strong conventional ways to do this, but there's a number of different possible ways that this goes into. We have things like current source density analysis. We also have actually doing the segmentation that we do with our 2 photon to get regions of interest. And then we have some higher order analyses like TCA, CBRA, GLMs, which you guys have already heard a little bit about today. But the most important thing I want to highlight today is this chapter here, which is the OpenScope experimental projects section. So for each of OpenScope's release data sets that we've released in the past, we also release associated notebook with them. So if you watched the presentation today that you found interesting, you want to dig into the data, I would encourage you to look at the associated notebook here. So these were created through collaboration with me and people on the external teams themselves. And the idea is this will help you actually do some basic analysis of the data and understand what the files actually are and what their purpose is. You have heard explanations of the experimental design and intent there, but this might serve as a little refresher. And I would use this as a sort of a jumping off point for people who are interested. And obviously, if you run this notebook, you find it interesting and you want to build upon it or investigate more, of course, we encourage you to contact us or the external teams and try to collaborate. Like I said, these these data sets will be released in the next coming months. So these notebooks don't currently work out of the box because the data is embargoed, but when it is unembargoed, these will be launchable and runnable with just the push of a button. And you shouldn't have to do a lot of work to try to get the environment set up or try to download the data. This should all be taken care of within the notebook itself. So I'm going to go over each of these. And then I'll just show you a brief example of how to actually launch these notebooks themselves to run them. So this is for our Vismo data set. You can go down here and there's sort of this metadata table we procedurally generated, which gives an overview of the data set itself. So you can see what files have, which mice, which genotypes, which stimulus types. And going down here, like I said, this will download the NWB for you. It'll open it for you, and then it will actually pull out the data. So this is like the segmentation masks from the actual imaged FOV. It pulls out the fluorescence here. You can see the fluorescence for the entire session. This produces a figure that you might have already seen in some presentations, which gives you a timeline of the session itself. And an important detail of the way we package the stimulus for Vismo is this is actually multiple overlapping stimulus tables. That's mentioned here, but just don't be confused if the colors seem a little bit like they're overlapping blocks here. This also gives you code to interpolate, to align to the stimulus and select stimulus times of interest. And then you can see we actually generate sort of trial-aligned responses of various cells. You can get tuning curves out of this and receptive fields. So, like I said, this is just useful if you want to just sort of get an understanding of what the data set itself is and how to use it for basic analyses, and you could build upon this. So for the texture data set, we have something very similar. We can see the textures here. Same thing, we have an experiment overview. This gives a mouse overview. And it does a lot of the same stuff. So it extracts the fluorescence, produces the same things. And then you can actually get a view of the stimulus times here, presented here for selecting. And then you get trial-aligned response windows as well. What's not shown here is what I mentioned a little bit earlier, which is there is behavior information in these files. These other notebooks describe how to get behavior information, and I'll show that in a little bit. But just because it's not shown here doesn't mean it's not in the data itself. So we also have a notebook for the loop data set, overview of the stimulus. This pulls out the epochs from the stimulus structure here. For this particular project, the stimuluses might be a little bit too complicated to represent just in one linear plot, but you get a sense for how it's sort of organized in terms of the fraction of time and the number of different types of presentations. And then this basically is doing more advanced analyses here. You can see the average sort of how trial-aligned responses are done here, but this is with ephys rather than ophys. And then this is a t-SNE model for clustering and classifying these responses. And then finally, we have our SciCode data set. Once again, it does a lot of the same stuff at the beginning. You can see here an overview of the probes that we actually used. We have another notebook which actually will pull from the data and get you the actual, for each file, it will get you the probe tracks for a data set. So that's referenced, I think, in some other notebooks. You can see the brain regions from the annotated coordinate space. And you can select those neurons associated here. So in this case, we select ones just from primary visual cortex and anteromedial. This does the same thing where it pulls out the stimulus epochs of the session, and you can get a sense for how it's structured. Also, the injection time as mentioned, or it's shown here. And one thing that I don't believe is encoded anywhere for the Psycho data set is whether or not the injection was saline or psilocybin. So Roberto mentioned in his presentation that, so there's 2 sessions for every mouse. The first one is saline and the second one is psilocybin. So that will be mentioned when we release the data set. It will be written in the metadata, but I don't think it's encoded anywhere in the files right now. So this notebook opens 2 NWs. It opens one for a saline and one for a psilocybin, and then we compare them. So you can generate the trial line spikes from the data here and get a matrix showing the responses. You can kind of sub-select response windows, and then you can also see this one is a psilocybin session. So you can compare the activity that way. And there's also the optotagging. So this kind of just shows how to pull out cells and get optotagging responses to the laser stimulus. So feel free to take a more thorough look at these notebooks if you're interested in any particular data set. And you should be able to find our contact information here in the webinar. But I also wanted to briefly just touch on our previous data sets. So we're releasing 4 this year, but in the past several years, we've released 3 each year. So we have, about a dozen data sets now. And I think there's a lot of really, really powerful analyses that could be done still on the data that is currently released. So like we have this one, which is the temporal barcoding data set, which we made with Pamela Reinigal. Vision to hippocampus, which we made with Chinmay Parandare and Mayank Mehta. And like I said, these ones are available. So, well, here's the Vision to hippocampus one right here. So I'd strongly encourage you to look at OpenScope's existing data sets. And I mentioned behavior data. So let me really quickly show you the behavior data here. One of these. Should be. Yeah, okay. So this notebook here pulls out the running speed here, which you can see, and the eye area. In addition to the eye area, there's also the eye coordinates, which are in a different notebook, but it is explained how to access that data here. So yeah, there's a lot of very rich, valuable data within all these data sets. And the data book, I think, will be a really great resource if you're interested in digging in any of that data. Now, finally, before I'm done talking about the data book, I want to explain how to actually use it. So if you're interested, you can, let's say, take this receptive fields notebook. You click this launch button here, and you can launch it onto a number of different platforms. I would recommend Jupyter Hub. So Jupyter Hub, this is actually Dandy Hub, which is hosted by Dandy. If you click that, there's a bunch of sign-in instructions. Okay, let's see. You can select your environment here. We want the normal Dandy. This usually takes a minute or so to launch. But what it's going to do is it's going to actually basically copy all of these notebooks into the environment here, and it should set up the appropriate environment. And then you should be able to just press one of the notebooks, basically run it, and then it will run and reproduce your analysis for you. So we did our best to eliminate a lot of the steps, which are, you have to sort of set up the environment. You have to sort of try to find the right data. You have to try to figure out how to open it, et cetera, et cetera. And we tried to streamline it all into just the push of 1 or 2 buttons in this case, to actually, get people to be able to quickly dive into the data and the analysis. So this is just going to take 1 more second. In the meantime, I'll also talk about contributions. So we have a tab here about how to contribute to the data book. we have all these instructions, so hopefully it should be pretty clear, but if it's not clear, you could contact me or somebody else. And then we also have what I briefly mentioned earlier, which is this contributors tab. So if you are a contributor to the databook, we have a number of external contributors, not just from these teams, but from other external teams as well. And anybody who has their analyses here gets added to this authors list, and then officially has authorship here. Oh, and I want to talk about reviews. So there is, we have pull requests, and there's this really nice tool called Review MB, where if someone tries to contribute to the data book, we leverage GitHub reviews in order to actually evaluate people's contributions and provide feedback. And there is a useful tool here, Review MB, which allows you to actually see what modifications have been made to the notebooks themselves. So I think the data book, it serves as a good example for a model that we could use for reproducible analysis, because I think data is essentially, it's not going the full 9 yards for reproducible analysis and collaborative science. You definitely need to be able to have code that's highly usable and well documented and easy to understand in order to reuse data if you want your data to be massively used. So this is going to take a few more minutes, but basically what will happen is it will open up a tab and you can just see a Jupyter Hub, and then you can easily run the notebooks and it will set up the environment for you, like I mentioned. So that is the whole OpenScope data book. Are there any questions?
Thanks, Carter. It's a wonderful presentation. Thank you.
Yeah, thanks.
Any question for Carter? Okay. It's overwhelmingly rich. But we try to make it really easy because, sharing data is a first order, but we want to do this with intent. So that's the goal of this session, is not only share data, but provide you with context. What is the experimental context of how we share the data? Who is the team behind it? So allow everyone to see the humans that are thinking hard and about the experiments and how to collaborate with them. And then what's the science, where they're currently thinking and what's their current focus. And then eventually how to access the data. So we provide code, and then there's metadata, right? So all of this comes with descriptions of the experiments and eventually plots just to get started. So we put a lot of intent into data reuse, and so because it's important. And so that's partly the goal of this event. I want to finish with the last slide, my talk, because I think it's important to highlight it. So let me share that. Can you see it? Okay. So as I said, Boost dataset are currently embargoed, but and we do this event ahead of time so that it's ready when embargo is lifted. So the first data that will be released is on May 6th, and then in the following weeks. And so this is essentially the time where the team had access to the a complete data set, but they could start the analysis in depth a year ago. And then on June 11, we released the data set from Loop, followed the following week by the texture project on June 17, and the Vismo project, which was a little more complicated to acquire and require more collaboration with the team initially. We'll release it on August 29. So that's the plan. Any question from the audience? Well, I want to thank all of the speakers for. Oh, yes, Javier?
Yeah, I'm just curious if maybe you can advance what's next for the OpenScope project for the following years. Are you going to do new calls, or are you going to focus on this predictive coding framework instead of collaborative research?
That's right. So we are working really, really hard to scale up the program, and it's just a couple months too early for me to speak about it. But in gross line, we think the project tracks that you apply is really fruitful. I mean, we've shown, in my mind, it's an amazing success. Obviously, I'm biased, but we've shown the process of selecting project, acquiring the data, and publishing. So the earlier project under NIH funding are not, I know, all knocking on the door of publication journals, and that takes time and effort, but we are helping the team, all of them, do that. And some of them made it. And so in my mind, it's a success. The community track is something that was unexpected. I didn't expect that coming in, and so we explore what's possible with this model. And it's been such a fun effort to collaborate with so many people on the shared experimental design. And so I think we should continue. And so what we envision is those two track to continue. So have focus project coming from individual teams, and then community synergizing effort around share questions. I mean, already today you saw that there share interest. And so I think there is a lot of potential in neuroscience for experiment that are designed jointly and serving many, many labs. I think it's a missing component in the field that would eventually blossom. And so we want to facilitate those two in the future. And so that, I mean, obviously that require support, and I'm incredibly thankful for your support and then other people's support in continuing the program. So that's where it's a bit too early, but we're working very hard on making this happen.
Yeah, no, thank you so much, Joram. I think I speak from everyone that it's been a wonderful experience for everybody, and that we've been learning a lot from each other. So it's been wonderful to keep these projects going.
I personally have grown as a scientist through it, so it's been fun. I've learned from all of you. Okay, I think we achieved our goals today. And so for everyone that is listening, please, if you're interested in one of us project, reach out to the team, try to continue the spirit of open science and collaborations. That's the goal. And as a field, we decide how we want to operate and how we want to bring people in. And so I think it's really important. And that's how I see it. It's really the goal is to solve the brain, not to achieve any particular paper. Okay, thank you, everyone. And I wish you all a safe and obvious day.
Thank you.
Thank you very much.
Thank
Thank
you.
you.
Bye.